There is a technical problem right now, so um, good evening, everyone, ladies and gentlemen, and fellow enthusiasts of the human mind. My name is İpek Köker. I'm a second-year medical student at Ankara University. And also, I am honored to serve as the head of the scientific team in Ankara University Neuroscience Community. As a community, we have been running a presentation series on our YouTube channel called Ankara Üniversitesi Sinir Bilim Topluluğu. And this series covers a wide range of topics related to neuroscience, and students from a variety of faculties, including psychology, biology, medicine, attend our presentations. And for those who wish to engage in discussions on neuroscience research, also we have established scientific and student groups um, where we collaborate with our professors and peers. And some of our key events also includes the Multidisciplinary Neuroscience Congress, Medical Technology Symposium, and Neurological Diseases Awareness Symposium. And today we are truly honored and excited to welcome you all and want to thank you all for attending our online presentation featuring Professor George Malliaras. And we want to thank him for being here with us today. We are truly privileged to have him here to share his knowledge and insights. And also I would like to thank my friends from neuroscience community who made it possible to carry out this event. Um, Before the presentation, I would like to briefly introduce him. George Malliaras is the principal professor of technology at the University of Cambridge. He leads the Bioelectronics Laboratory, an interdisciplinary group of scientists, engineers, and clinicians who translate advances in electronics to better tools for healthcare. George Malliaras received a BS from the Aristotle University, Greece, a PhD from the University of Groningen, Netherlands, and did a postdoc at the IBM Almaden Research Center, USA. Before joining Cambridge, he was a faculty member at Cornell University in the USA, where he also served as the director of the Cornell Nanoscale Facility and at the School of Mines of Saint Etienne in France. His research has been recognized with awards from the European Academy of Sciences, the Materials Research Society, the New York Academy of Sciences, the US National Science Foundation, and DuPont. He was awarded an honorary doctorate from the University of Linköping, an elected fellow of the Materials Research Society, and of the Royal Society of Chemistry, and he is a member of the Academy European and European Academy of Sciences. He serves as a deputy editor of Science Advances, and he is the director of the EPSRC IVS in targeted delivery for hard to treat cancers. And today we have the distinct privilege of welcoming him here to share his wisdom with us. And also, I would like to encourage all to think this presentation as an amazing opportunity to expand your horizon on neuroscience. You can ask questions by filling the question form. And without further ado, I close my microphone and leave the floor to Professor George Malliaras.
Thank you very much for the kind invitation. The pleasure and the privilege is all mine. It's an honor to be invited to present. I'm delighted for this, and I thank you very much. So I'll give my talk. I'll keep it to about 45 minutes. Then I look forward to a discussion, questions. I'm super happy to be engaging with this audience. Let me share my slides, and please tell me if you can see the slides and if they're on full screen. Give me 1 second to do that. All right, so is this full screen?
I'm so sorry for interrupting you, but we have a technical problem. So can we just can you just please start the presentation after 3 minutes?
Yeah, sure, absolutely. No, not a problem at all.
Thank you very much. Thank you very much.
Do you see my slides? Yes, I have a message here saying yes. Perfect. So I'll wait. Let me know. Give me the okay when. actually, I'll take this opportunity to go and remind my kids to be a little bit quiet. They just came back home from school and they're very excited. So I'll go do that while we're waiting. Is that all right?
Okay, it is all right. Thank you very
Right.
much
See
for
you
your
in a moment.
understanding. Okay, see you.
Great.
Okay, we will be glad if you could start the presentation. And thank you very much for.
Yeah, excellent. Good to go. So welcome everyone. Thank you so much for taking the time to attend my presentation. My name is George Malliaras. I'm at the University of Cambridge in England. It's dark and it's cold out here. I hope it's warmer where you are. What I wanted to discuss today is technology for bioelectronic medicine. I will give you my view of what bioelectronic medicine is, how it's evolving, and why it needs new technology, and then discuss some work, mostly from our group, to address this issue. So let me start by giving you my understanding of what medicine is. I'm not a medic. I was trained as a physicist, then switched to engineering. And it goes as follows. So as we go through the journey between birth and death, the length of that journey is mostly determined by our genetic material that we inherited from our parents. There are factors, however, that can accelerate this journey. A poor lifestyle can send people to an early grave. Pathogens, very much in discussion after the COVID pandemic. Accidents can all serve to decrease our life. And in this context, medicine is the set of tools, the know-how that we as humankind have developed to turn back the clock, basically a decelerating mechanism that gives us more life and more quality life. How does medicine do that? There are a variety of modes of intervention, starting with the most classical one, which is pharmaceuticals. They can be used for prevention, for example, vaccines. Again, very topical subject after the pandemic. You get vaccinated to prevent you from getting sick. They can be used for symptoms control. For example, the administration of dopamine in people with Parkinson's disease can control the symptoms. It doesn't cure the disease, but it's symptoms control, and can also be used for curative outcomes. For example, the administration of antibiotics will kill the bacteria that cause disease and cure the patient. In the same vein, surgery, radiotherapy, when you intervene and cut and burn and reconnect tissues has been used as a classical mode of intervention with a variety of outcomes. So, for example, the removal of a breast in people who are at risk of developing breast cancer is done for prevention. The installation of a shunt, a tube between the brain and the abdominal cavity can drain excess cerebrospinal fluid and help control the symptoms of hydrocephalus. And the resection of a tumor before it has had the chance to metastasize can cure the patient of cancer. In these classical modalities, there are two new ones that have been added in recent years. One is regenerative medicine, where you go into the body and add tissues, usually produced through stem cell techniques. And this has been shown to have excellent outcomes, curative outcomes, in people, for example, with leukemia. The transplantation of stem cells can provide a curative outcome. In the same vein, bioelectronic medicine, you go into the body and add an electronic device, which through electrical stimulation can endow a variety of effects. For example, it can be used for prevention. This is the case of an implantable cardioverter defibrillator, which is a device that has been recently in the news. This was about 2 years ago during a football tournament where a footballer from the Danish team collapsed in the field because he suffered cardiac arrest. And he's now fitted with this device. This is a little kit of electronics that contains a battery and different circuits here, and is connected through electrodes to the heart. It monitors the beating of the heart, and if the heart stops, if the patient suffers cardiac arrest, it will apply a high current pulse and defibrillate and get the heart starting again. And Eriksson, the footballer who's now fitted with this device, plays for the Premier League in the UK. So this is an example of prevention of death through defibrillation. Having said that, most bioelectronic medicine interventions today are in this space. They're there to do symptoms control. An example is deep brain stimulation for tremor. This is shown here in this video, which should hopefully start soon. So this is a patient who suffers from a movement disorder that is called tremor and is characterized by uncontrolled movement of the extremities. And you see, it makes life difficult. They cannot write, they cannot hold a glass of water without spilling. And through electrical stimulation of the subthalamic nucleus using an implanted electrode array, you can control the symptoms. You have a significant amelioration of the symptoms that allows people to live almost normal lives. So this deep brain stimulation relies, as I mentioned earlier, on implantable devices that are connected with two electrodes that are placed in the brain in this case. And through electrical stimulation, you can control the symptoms of movement disorders, such as dystonia, Parkinson's disease, tremor. You can control the frequency and intensity of seizures in epilepsy. They have been recently shown to have beneficial effects in severe cases of obsessive compulsive disorder and depression. They're being trialed for obesity and a variety of other conditions. Now, in the ancient world, electric fish were used to deliver an electric shock to the head to help people with migraine. It's not clear how well that intervention worked, but the people were already using electricity for therapeutic effects. It was Galvani in the 18th century that started putting this phenomena on a more scientific basis. The famous experiment where he took a dead frog and applied electricity and got the legs to twitch. And then 200 years later, there were medical devices. This is a pacemaker. The year before these devices became fully implantable. And at that time, the patient had to carry with them at all times this life-saving cart of batteries and electronics. And then if we start the clock at 1958, which was the year when the first fully implanted pacemaker was developed, it was developed at the Karolinska Hospital in Sweden to treat cardiac arrhythmia. So here, the location of the intervention is on the heart. So we start, excuse me, we start the clock at late 50s, then late 60s. There were already cochlear implants for helping people with profound deafness. So the location of the intervention here is the inner ear. A decade later, late 70s, there were spinal cord stimulators for helping people with chronic pain. Then a couple of other peripheral nerve stimulation devices. In 2000, the brain came to the forefront with deep brain stimulation for Parkinson's, neurological, and then neuropsychiatric disorders. And then it exploded. Lots of devices. This is not a comprehensive chart. The latest and greatest is peripheral nerve stimulation for expanding the scope of bioelectronic medicine beyond neurological and into autoimmune diseases. For example, Crohn's disease, type 1 diabetes, rheumatoid arthritis are believed to be treatable with this technology. It has gotten to be significant enough so that companies such as Google's parent company Alphabet, and GSK, a major pharmaceutical, got into the game through a partnership which gave rise to a company called Galvani Bioelectronics here in the UK that aims to treat disease through miniature implants that stimulate peripheral nerves. So a big step away from pharmaceuticals. And at the same time, the founder of Tesla, of SpaceX, the founder of Facebook, have all been involved in this game. There is a lot of hype as a result. If you look into Hollywood movies, for example, it's always a bad person that wears bioelectronically controlled tentacles that tries to destroy our civilization. This is the image that is portrayed. However, the clinical reality is more what you see here. This is a boy who wears a cochlear implant. This is a device that consists of an external part that has a microphone that picks up the sound, then some electronics that process this sound, and an antenna that radios that information into an implant in the inner ear. And through electrical stimulation in the cochlea, which is in the inner ear, this boy can hear for the first time in his life. And just look at the wonder in his eyes as he's able to do that. So we're talking about life-changing technology. Now, any medical technology raises ethical questions. Here in bioelectronic medicine, we are often acting on the brain, which is the organ that gives us sense of self, is the seat of consciousness. So as a result, the ethical issues are compounded and are quite unique. This caricature here shows the quintessential mad scientist working in an ivory tower away from society, trying to galvanize, as it was called at the time, or reanimate by using electricity, a dead corpse. And as a result, this is something that society had a very strong reaction against at the very early years of bioelectronics following Galvani's work. Now, how did we arrive to this life-saving technology? Through continuous interaction with the stakeholders in this game. So the message I'm trying to pass here is when you're developing medical technology, it's very important from the very beginnings of this endeavor to engage with patients, with their advocates, with ethicists, with regulators, with industry, with a continuum of stakeholders in order to be able to produce such miracles of technology. Now, with that as a very broad introduction to bioelectronic medicine, let me start now getting a little bit more technical and more into the detail. This is from a recent paper that surveys different implants that are available today, their location, and the conditions they are meant to treat. And you see implants in different parts of the body and different conditions that can be potentially treated. And by and large, these implants have a structure where you have an implantable pulse generator, as it's called. It's like a tin can that contains a battery. About 2/3 of its volume is the battery. And then about 1/3 of its volume is electronics. And then there is a connector with extension wires, one level of connectivity here as well, and then leading to an electrode array with different stimulation sites or different electrodes. In this case, there are 2 of those because for deep brain stimulation, we do a bilateral implantation targeting structures at both hemispheres. So again, here you see the electrode arrays implanted in the subthalamic nucleus, or at least one of the electrodes is, and then cables with slack with a level of connectivity here, and cables here tunneled under the neck to the chest under the skin where this implantable pulse generator is placed. If you look at the very front end of this device where electrodes interface with tissue, it looks very similar across all these devices. So this is the electrode array of a cochlear implant. It is shaped like this because the cochlea looks like a snail, so it has to follow its curvature. These are 2 different technologies for spinal cord implants, a so-called paddle electrode, the 2-dimensional array, and then a percutaneous 1-dimensional array. And by and large, what you see is chunks of metal that are either cylindrical or flat or curved, and they're made out of platinum with a little bit of iridium to adjust the mechanical properties. And they're encased in an insulator, shown here, shown there, which is either a urethane or a silicone that holds the structure together and provides a level of insulation to the wires that come from the electrode to the outside part. These are made by hand. So if you go to Medel, Medtronic, Boston Scientific, their fabrication facility looks like this. It's people working under through microscopes, and they manually assemble these devices, which is incredible. This is watchmaker technology. Back from the times of old, when mechanical watches were assembled in Switzerland using these techniques. In microelectronics, we have better ways of making electronic devices. This is TSMC, Taiwan Semiconductor Manufacturing Corporation. It's a factory that runs almost fully automated with very few people around, and using processes such as deposition of thin films in vacuum, photolithography or beam lithography, etching, you can produce objects of exceptional precision, incredible throughput, amazing reproducibility, and these are properties that can have tremendous benefit to bioelectronic medicine. So we will call this thin film implants. By that, I mean implants that are made using this technology from the microelectronics industry. And we'll talk about their property. So here is an example from our own work. We make very thin, flexible implants that have microfabricated electrodes. These are approximately the size of the soma of a single neuron, so of the order of tens of microns, micrometers. These are meant for cortical usage, so either sitting at the surface of the brain, the spinal cord, be wrapped around nerves, but they're also thin film implants that are implantable. This is an example from Tesla, from Elon Musk's company called Neuralink, where they microfabricate this needle-shaped implants. They have a suture loop here at the bottom, and there is a surgical robot that will pick those up and push them into the brain, as shown here. And these again have microfabricated electrodes of the size of a single neuron. And they just got approval by the FDA to proceed to human trials. So these are slowly entering the human use, and it's a very exciting time. So what I want to do is to start with this manufacturing technology and look at properties that can be obtained when we use this technology. These are properties at the on the level of an implant, and then look at potential medical benefits that can be obtained. So let's start with the first one, which is, as long as you make implants very thin, then they can become flexible or even stretchable. This has been demonstrated by a couple of groups. One example is a group of John Rogers in the US, who have made very thin implants on a sheet of plastic called polymethyl methacrylate. So thin, actually, that if you lift it up to put it on the brain, it will collapse under its own weight. So what they do is they pour silk, a solution of silk, on the implant, let it dry, so you have a layer of silk on top of the implant that gives it temporary mechanical strength. And then the surgeon can pick up the implant, place it on the brain of the patient, and then dissolve using saline the silk. And what is left behind is a tattoo-like structure that intimately conforms to the curvature of the brain. So you have excellent mechanical contact with something which is almost imperceptible at the surface of the brain. Stephanie Lacour at EPFL in Switzerland took this a step further. She uses elastomers, such as silicone rubbers, to develop electronics on these materials. And as a result, the electrode arrays that she develops can stretch and thereby follow movement in parts of the anatomy that tend to move when the animal behaves. So in this case, the spinal cord can undergo motions, changes of dimensions of the order of 10 or 20%. And these implants can still keep good mechanical contact even under these extreme conditions. So what this flexibility and stretchability give you is better apposition, better contact to tissue, good mechanical properties. And also when you put them in the brain, because they're very thin, they tend to elicit reduced scarring. I didn't show you this data, but this is indeed the case. Another advantage you can get with this technology is miniaturization. You can make very small electrodes, as I've already shown you. However, as we make electrodes smaller, their ability to record or stimulate diminishes, simply because the interaction between electronic and ionic carriers is very weak. Here, you need to do one more trick. You need to introduce a material that will allow this interaction between electronic carriers in the electrode and ionic carriers in the brain, sodium, potassium, what have you, to couple a bit better. One example is a conducting polymer called P.PSS. We're not going to go into details of the structure. Consider this as a spongy conductor where ions from the cerebrospinal fluid, sodium, potassium, chlorine, or so, can penetrate into the structure of this film and couple with electronic charges. As a result, this gives you a much stronger interaction and allows you to make very small electrodes that can both record and stimulate very efficiently. So with this technology, we can go from very large electrodes that are used currently in the clinic to map the brain to very small ones that, again, can be used in the clinic to map the brain at exceptional precision. What you're seeing here is a cortical grid with 9 electrodes, handmade, and the electrodes here have a diameter of 5 millimeters. This is placed under the dura, so you see a craniotomy here, you see a durectomy, the dura is folded back here, and the array is placed on the cortex to map, provide functional mapping of the brain before resection surgery. Here you see one of these microfabricated arrays where you have hundreds of electrodes in the same space, providing mapping down to a single neuron level. These arrays have been shown to measure single neuron activity from cortical layers and can give exceptionally precise maps, helping to guide the resection at much higher precision than the current clinical standard. In addition, by microfabricating electrodes, you can also precisely stimulate. This is an experiment where a 3 electrode array device was developed with micron size, again, microelectrodes, and implanted on the nerves of the branchial plexus that innervate the muscles of the hand and are responsible for controlling hand motion. Through precise stimulation with these electrodes, we can recover a maximum degree of motion, of articulation in an animal model. So this is important. Why? Because imagine that you have an injury at the spinal cord or the nerve that disconnects the muscle from the brain. So the brain is firing commands, and those commands do not convey to the muscle. What you could do is you could stimulate the nerves here and obtain the required motion. So through miniaturization, you can get more precise recording and stimulation of tissues. Another property of thin film implants is multimodality. What do I mean by this? You can endow properties such as, for example, optical transparency, because these implants are very thin. They can be made optically transparent so that you can have electrodes through which you can shine a high-intensity laser beam without inducing any noise during recording, and at the same time do super resolution microscopy to image, for example, markers in the synapse with molecular scale resolution. At the same time, you can make them also compatible to MRI and combine electrophysiology, optical techniques, and structural and functional MRI, thereby collecting more information about the brain. You can introduce different sensors. This is an implantable array that penetrates into the brain and contains electrodes that measure electrophysiology, together with transistor-based sensors that are functionalized, for example, for measuring lactate. And what you see here is a simultaneous measurement of electrical activity. So this is a series of seizures that grow closer to each other, more frequent and more intense. And at the same time, in the same location, you can get trends in metabolic activity. You see that lactate increases every time there is a seizure. You take a distinct step up in lactate, indicating that the brain locally gets tired. And similarly, ion sensors are being developed to the point where they're getting ready for implantation in the brain to follow this phenomena at the appropriate time scale. Drug delivery devices can also be made with this technology. This is an example of a microfluidic device that was developed together with two electrodes shown here. So this has a fluidic channel through which you can bring into the brain a solution of a drug. And then once it reaches the delivery point here, you have holes that are capped with an electrophoretic membrane. And when you apply a potential, you can get the drug from the solution to come out into the brain with good spatiotemporal control. So what you can do with this, you can stop or even prevent epileptic seizures, as shown here in an animal model, by delivering an inhibitory neurotransmitter such as GABA. So what you see here is the device is implanted, and through administration of 4-AP, a series of seizures is elicited. Here is the same experiment, and when the seizures commence, you turn the device on. It gives a couple of puffs of GABA, and the seizures are immediately gone. And here is an experiment where you turn the device on prophylactically, and you try to induce a seizure, and just the seizure cannot materialize because you have biochemically prevented hyperexcitability. So these multimodality allows you to get more information about the status of the brain, both in physiology and in pathology, and permits combination therapies, for example, electrical stimulation together with biochemical stimulation. Another feature of these implants is that they can be shape actuated. As they're very thin, they're quite straightforward to change shape. And this brings us to the interface with the field of soft robotics. This is one example here. This is from George Whiteside's group at Harvard, where they use pneumatic actuation. So they use compressed air or fluid to change the shape of a soft object. As a result of this change of shape, this object can move. But the important thing here is the change of shape. And this can come in handy in medical applications. This is one example. And it comes from spinal cord stimulation. So stimulation of the spinal cord is an established neuromodulation technique since, again, the late 70s or so, used to treat chronic pain. Electrical stimulation can cause the painful sensations to be replaced with more agreeable ones and is used clinically for that purpose. There are two technologies. One is a percutaneous array, which is a linear array with cylindrical electrodes that is placed on the spinal cord through the space between vertebrae. So here, an anesthesiologist will feel the space between vertebrae, jab a needle in there, and pipe that device in through the needle in a similar fashion that an epidural injection is given, or a spinal tap is performed. So you do you find the space and you can access it percutaneously. So the placement of the array is very facile. The patient goes home a few hours after the operation. However, this array addresses only a very small part of the spinal cord and can move out of alignment as the patient moves. So the disadvantage is this device might lose efficacy altogether, and then revision operation is required. Better technology is this two-dimensional array. This is called a paddle electrode, and it has many more electrodes distributed over an area. So when you place it against the spinal cord, you can always find a good area to stimulate and achieve efficacy. It also doesn't move. Once placed in, it stays there. However, it requires a laminectomy to place. You need to remove part of the vertebrae, and this is associated with a lengthy hospital stay. The patient has to stay at the hospital to recover from this, which increases cost. So patients have a difficult choice to make: either a device that requires a minimally invasive procedure but might lose efficacy altogether, or a device that requires considerable surgery but it's very efficient. So here we took inspiration from soft robotics and developed thin arrays with embedded microfluidics. You can roll those to the shape of a percutaneous array and then expand it into the body through pneumatic actuation. So the idea is as follows, is that you do a lumbar puncture, and then you put the device in the shape of a percutaneous array, and then expand it once it has reached the location where the intervention should take place. This is shown here. This is the lumbar puncture here. And this is the device being inserted on the spinal cord. Here is a fluoroscopy machine that through gentle X-ray illumination can localize the device. And sorry, this is the device being actuated post-implantation. And here you see the device coming in and being placed. There are two X-ray markers at the top and bottom part of the device that coincide when the device is rolled up, but then they begin to separate when it gets expanded. So this technology now is approaching first in human stage, and hopefully will be placed in a living patient soon. Another way to actuate, oh, sorry, I forgot to that I had this slide. So this is where the device was injected, and here is a laminectomy that shows that the device really conforms nicely to the spinal cord. And then this thing here and this thing here are the X-ray markers that show positioning. Now, another way to actuate is through electromechanical actuation. Here you rely on having a polymer, very thin polymer film in contact with a thin metal film. And when you apply a potential, ions can come from the solution into the polymer, swell it, and that deforms the structure in this fashion. Or if you pull the ions out, then you deform in the other fashion. You can get significant motion as it's shown here. This is a real-time video of one of these devices doing its trick. And again, through this pulsing of a voltage, you can repeatedly change the angle of actuation. And you can do this over and over and over as a function of time. Now, this is a video that shows that you can engineer the type of motion. You can make it go helicoidally around a structure. And this you do by tilting a little bit through microfabrication the slant of the polymer structures with respect to the axis of the device. And with this motion, you can wrap around a nerve, as shown here. And if you introduce microelectrodes, then you can gently wrap around a nerve and then monitor its health. This is done intraoperatively. For example, when a surgeon operates in an area that has a lot of nerves, they should monitor nerve health to make sure they don't cut where they shouldn't. This is done typically with a device that is very invasive to the nerve. So this wrapping around the nerve gently can provide non-invasive monitoring. So this shape actuation leads to less invasive insertion and expansion of devices and less invasive monitoring of different phenomena. Now, the final thing I want to talk to you about is biointegration. What do I mean by this? This brings us to the interface with the regenerative medicine. As I mentioned earlier, this field involves taking cells from a patient, bringing them to a pluripotent stem cell state, and then forward differentiating them to different cell types, for example, neurons, muscle, what have you, and then putting them back into the patient in order to undo a deficit because of injury or disease. Here, we're talking about combining these two approaches, combining the implantation of cells, of tissues, with the implantation of bioelectronic device. So we're in this space that we're gonna call regenerative bioelectronics. And the argument goes as follows, that you're bringing now an implant that contains electrodes, but also tissue. And you hope that when you bring this into the body, you'll have some re-innervation. Either the tissue that you bring will innervate the host tissue or the other way around. So you'll have a regenerative component that will first and foremost camouflage your electronics from the body, so it increases biocompatibility. And then it will effectuate a tissue repair through this reinnervation and enhance communication between host tissue and implant. And at the same time, the bioelectronic component can monitor this process of repair, can even guide it, and at the end, it will control repaired tissue. So we use stem cells that are derived from fibroblasts and then forward programmed using a process which is commercial. This is technology developed by a colleague in clinical neurosciences called Mark Cotter and has been commercialized by a company called Bio. What you see here on the video is this forward differentiation process that turns these pluripotent stem cells into myocytes and then into myotubes, giving rise to nice muscle structure that can then be implanted. So to prove this concept, we work on a peripheral nerve injury model. The idea, again, is if you have a peripheral nerve injury, the brain gets disconnected from muscle. And if you're able to record at the stub, which is proximal to the nerve, you can pick up this motor information and then use it to stimulate, as I showed you earlier, past the injury site and recuperate that motion. But recording from severed nerves is exceptionally difficult. So here we hypothesized that if we provide an implant that has muscle on it and we present it to a severed nerve, then the nerve axons will innervate that muscle. And every time the brain fires with a motor command, that will be conveyed down the axon conduit and then stimulate the muscle cells. And we can pick that up in the form of a miniature myogram and then obtain the volition for motion. So the protocol goes as follows. We take a flexible array, seed it with the cells, forward differentiate them ex vivo, and then once we get this myotube formation, present it to a severed nerve and hoping we'll get the innervation. We did histology to make sure the cells remain alive, but this is the important thing. Here is an array without any tissue, just an array presented on the severed nerve. And what you see is when you stimulate the nerve, you only pick up the stimulation artifact. The reason for this is the nerve degenerates and ceases to convey any information. That's why measuring from severed nerves is very difficult. However, if you put muscle on the implant and then you present it here, then this reinnervation takes place and it maintains nerve health. So if we stimulate it now, you can measure this evoked potential-like structure, which is similar to what you measure in an untransected nerve. And then these are recordings of spontaneous activity, week 1, 2, 3, and 4 post-implantation. And you see that after week 3, you start seeing muscle activity being recorded, shown very clearly here, showing that successful integration takes about 4 weeks. Now, if you scale this data up to 1 and correlate them with behavior, these are epochs where the animal was moving, you see a correlation which is larger than the, higher than the statistically insignificant point, than the random point. So it is statistically significant correlation between motion and recording at week 4, showing that indeed successful innervation has taken place. So this is a very interesting concept that shows how the synergy between two fields can lead to new ways to treat untreatable conditions, such as, for example, peripheral nerve injury. So better integration with tissue and tissue repair are the advantages. That sounds super exciting. Look at all these properties, look at all these benefits that you would have. What's the catch? Why is this not rolled out yet? There are some formidable challenges. First and foremost, stability, which relates to the safety of the intervention. So the devices have to be proven to be stable over a long term. And because they're very thin, any defect, any instability will immediately lead to catastrophic failures. So to investigate this, we have set up an ISO-inspired accelerated testing platform where we take implants, we stimulate excessively in media that is kept at 70 degrees. So we have a 10x acceleration in terms of time scale. We spike the media with hydrogen peroxide to make it reactive and stimulate the environment after implantation. And we also agitate to simulate mechanical motions of the implant. And then we do implants with different architectures, never mind the structure here. And then do failure mode analysis, looking at phenomena such as delamination of the electrode, corrosion of the electrode. By measuring amount of material in the electrolyte, we can tell how much of the electrode has decayed. And through these studies, we went from implants that barely last a year inside the body to implants that seem to be very stable. This is still running, but it shows an extrapolated lifetime pushing at least a decade. So progress is being made on this front. Another issue is X-ray visibility. If you make them thin, they're not visible to X-rays. They don't scatter much. So here we're developing markers using bismuth or barium salts that are compatible with lithographic processing and can be introduced in these devices. Other issues involve implantability, explantability. Being very thin, they require special procedures to put in. You need to be careful when you take them out to make sure they don't break. Specialty circuits, because you have now many electrodes, you have different devices. Packaging, how you connect these flexible arrays with more rigid structures such as circuits. Lots of issues to be addressed. But the conclusion is that the adoption of microfabrication techniques enables implants with an amazing array of properties that translate to significant clinical benefits for bioelectronic medicine. And there are formidable challenges that remain to be addressed, but the future looks very bright. So with that, let me thank my group. The group is co-led by a neurosurgeon and a neuroscientist, and it contains the continuum of expertise from physics, chemistry to neuroscience to surgery, and collaborations in Cambridge and outside, funding agencies. And let me thank you for your attention, and I'm ready. And of course, first and foremost, thank you for the kind invitation. I am now ready to take your questions.
Thank you very much for this amazing presentation. We're really grateful for this experience. There is I'm waiting for the questions, but before that, I would like to give you a minute. I would like to give you the certificate for this amazing talk. Thank you very much.
Thank you. Thank you. And please feel free to email me if there is a question that pops up later today. Just put it in an email. I'll be happy to engage through email.
Dear participants, if you have any questions, you can turn on your microphone and ask the questions. Actually, there is one question. What are your thoughts about Elon Musk's Neuralink?
Sorry, say that again.
What are your thoughts about Elon Musk's Neuralink?
It is very controversial. And they are driving forward the technology, and the fact that they made devices and made them approved by the FDA in such a short time is impressive. They are targeting people with quadriplegia, people who have typically spinal cord injury at a high level close to the brain. And they aim to restore communication capability by recording from the motor cortex. It's all wonderful. However, you have to be very careful when you develop medical technology to manage properly expectations. If you make exaggerated claims, at the end, it comes back to hurt the whole field. So where everybody in the community is a bit excited, but also a bit nervous, hoping that they will not rush into anything and do things at the appropriate pace. It is great to have a large investment, private investment in the field, but it has to be managed right. Otherwise, it can hurt the field for many years to come.
Thank you very much for your response. I guess there is no questions, but we would like to take a picture with you, and any participants who can turn off their cameras also can join us. I'm waiting for them to open the I will take the picture in 5 seconds if there is no other person who can turn on their cameras. Yeah. Thank you very much for this amazing talk. We are truly enjoyed and really delighted to host you today. It was really amazing, and thank you for your contributions to the growth of our community.
Thank you. Thank you for the invitation. And again, if there are any questions, just shoot me an email. I'd be happy to engage.
Thank you very much. You're really kind, and we're really grateful for this presentation today. I wish you a good day.
Thank you. Have a good evening.
Night.
Bye-bye. Bye.
Good evening.